Hello and Assalamu alaikum. Wishing you happy and healthy life. Today we will discuss about the role of bacteria in the process of the biodegradation. And there is a relationship between the microbes and the process of the bioremediation. We want to clean our environment, or we want to remedify our contaminated environment with the help of the biological sources. Now, question is that how bacteria degrade or decompose contaminated organic or inorganic compounds that are present in the contaminated air, contaminated soil, contaminated water due to oil spills and other industrial effluents, and so many contaminations due to industrial um, flow. Anyhow, because bacteria have the specific system of enzymes. Enzymes of the bacteria are their main metabolic machinery. Through these enzymes, bacteria and other microbes degrade or decompose organic or inorganic materials. Basically, bacteria are natural decomposer. We know about the phenomena of the decomposition, but natural degradation or decomposition process is very slow, at very slow rate. No. What are the bacterial enzymes? Enzymes are protein in nature, and genes are responsible for encoding of protein synthesis. And we know about the phenomena one gene and one protein phenomena. It means there is one gene for the encoding of the single enzymes. Specific genes are present for each type of the enzyme. Now, it has been reported through the different researchers, through different scientists, that some of the genes that were responsible for the encoding of the enzyme with biodegradation potential were plasmid based. What is meant by this? It means the enzymes encoding genes were present in the plasmid, and those genes were responsible for the production of those enzymes with degradation potential. Now, we know about the plasmid, that plasmid are extra chromosomal circular DNA that are present in the bacteria. And exchange of the plasmid from one bacteria to another bacteria is possible to advance molecular techniques like gene manipulation, cloning, etc. In this way, bacteria having no plasmid can be produced into the bacteria with plasmid and utilized in the biodegradation process. Through advanced techniques, number of the bacteria with degradation potential can be enhanced by exchange of the plasmid to other bacteria. Biodegradation can be done through the two approaches or techniques, one by using the pure culture and another one is by using the mixed microbial culture. What is the difference between the pure culture and mixed culture? Pure culture means having the single type of bacteria or one type of bacteria. Look at this diagram. This plate have the single types of the bacteria. It may be Bacillus, Pseudomonas, or um, any bacteria that is responsible for the degradation of the contaminated compounds. And look at this plate. This plate consists of the different types of the bacteria, and this is called the mixed culture. This is also, you know about the word consortium and consortia. Consortium is a singular, consortia is a plural word, or in simple words, you can say it is a group of the bacteria. When we talk about the process of the biodegradation, mostly consortia or the group of the bacteria are more preferable for the complete degradation. Why? What is meant by the complete degradation? We will discuss in the next slide. Look at this slide. It is representing the incomplete and complete degradation process. This is a compound that we want to degrade with the help of the bacteria. Suppose there is a single type of bacteria, or we are using the pure cultures to degrade this compound, and this single or single bacteria or pure culture convert this compound into the intermediate compound, and no further bacterial strain or bacteria are present for the further degradation of this intermediate compound. Suppose if this intermediate compound is toxic, even then more toxic or less toxic, but if it is toxic in nature in this intermediate compound, and there are no bacteria present to further decompose this intermediate compound, then it will remain in that environment or in that habitat and will get accumulated, and this will be danger for the environment. Now look at the second example. We want to degrade this compound with the help of the microorganism. Here are there are the different types of the bacteria. Bacteria A, B, C, and D, and each bacteria is uh, involved in the degradation of the first step, then next step, then next step, and next step. It is a sequential, we can say that it is a sequential degradation, and at the end, compound is completely degraded with the help of the different types of the bacteria or with the help of the group of the bacteria. And now this is called the complete degradation, and it is representing the incomplete degradation. This is favorable for the environment because when the compounds are completely transformed or converted into the inorganic form, actually, in this way, we can clean the environment. And now look at this. Each bacteria has its role for every step, and in this way, by the degradation of these compounds with the help of the bacteria, these bacteria can get their energy for their survival, for their growth, and secondly, they can metabolize or they can decompose this compound completely into the inorganic form. Now the process of the biodegradation can be speeded up by using the two ways. One is called the biostimulation and second one is called bioaugmentation. What is the difference between these two techniques or ways or approaches? Biostimulation means by activating existing microbes through providing favorable conditions like optimum pH, temperature, nutrients, air, proper water content, and so many favorable conditions. When we will provide these favorable conditions towards the existing microbes in that habitat, or we can say in a simply the indigenous microbes that were degrading the toxic compounds at a very slow rate, but when we will provide the favorable conditions, they become active, and through this stimulation, the biodegradation process will be enhanced. Second approach is by using the augmentation approach or way. Its mean addition of the external microbes with great potential of the degradation to speed up the degradation process. What is meant by the external microbes? It means if we have a genetically engineered microbes or we have isolated and other bacteria or other types of bacteria from different habitats or from different environments, and those bacteria have a very good potential for the degradation process, then we can introduce in the contaminated environment, and there are the already existing microbes, those who are degrading the contaminated compounds at a very slow rate, but we provided the favorable condition plus we provided or we added the external microbes to overall enhance the process of the degradation. Okay, dear students, this was all about the role of the bacteria and the biodegradation and how bacteria can actually what is the phenomena behind the biodegradation. It is the enzymatic system that is responsible for the degradation, and by utilizing the microbial enzyme we can enhance and we can convert the contaminated environment into clean environment by using their metabolic machinery, that is microbial enzyme, bacterial enzyme, and there are the different types of the enzymes they release surrounding their environment. Inshallah, in next lecture. We will discuss some other topic related to the bioremediation, microorganism. Till then, bye. Allah Hafiz.